Hi everyone, my name is Sheila Fitzgerald and I'm a fourth year PhD student in the Department of Chemistry at RCSI under the supervision of Professor Donal O'Shea. Today I'll be talking to you about some of my work on bioconjugations, inflow for drug conjugates and theranostic imaging agents. My work focuses on bioconjugation strategies, essentially taking a therapeutic biomolecule and tagging it with a near-infrared fluorophore to generate these targeted drug hybrids with dual functionality. So you have both a therapeutic biomolecule and a diagnostic imaging agent in one single theranostic entity. So we try to do this through the use of highly efficient chemistries, all the while maintaining mild flow conditions that will be transferable to a wide range of biomolecules. We also looked at developing an intra-chip reaction analysis system by taking advantage of the narrow channel sizes here, so they measure about 250 micron in diameter. One of the near-infrared fluorophores I've been working with is this Pyronitrophenyl tetrazine derivative. So the tetrazine portion of this fluorophore causes excited state quenching of fluorescence, but once it's reacted with a strained alkyne through an inverse electron demand Steel-Alder reaction, the fluorescence is actually switched on as the product is formed. For the purpose of this poster presentation, I'm going to focus on the cyclic RGD BCN peptide as my biomolecule. Why we chose RGD was because RGD has the ability to target transmembrane proteins on cancer cells, and also a number of RGD derivatives have undergone clinical trials as tumour therapies and diagnostic imaging agents. Optimization of this reaction was carried out in terms of time and temperature, with optimal conditions being identified as 40 degrees Celsius and a 5-minute residence time in the chip at equimolar concentrations, which then also removed any purification requirements. So this was done by fixing an excitation source and near-infrared fluorescence camera directly above the chip, which allowed us to do real-time image analysis in terms of fluorescence. So as the starting materials entered the chip, the fluorescence was quenched, but as the reaction proceeded and the product was formed, the fluorescence began to increase across the chip. So the chip was broken into 6 regions for monitoring, and as we can see here, increased rapidly in the early portions of the chip, but as the reaction began to reach completion, the fluorescence began to reach a maximum plateau across the top of the chip. The photophysical properties of the product can be seen here, and they show that it's an ideal candidate for near-infrared imaging. And we can see here an example of in vitro imaging showing the membrane stain of the cell, which is typically what we would look for when we're looking to do tumour imaging. So just to sum up, we have developed a method for generating novel drug hybrids with optimal conditions for biomolecules found to be just a 5-minute reaction time at 40 degrees Celsius. And we have also developed a system for intra-chip reaction monitoring. I'd just like to thank my supervisor, Professor Donal O'Shea, and also Dr. Dan Rue and SSPC for all their financial support. And thank you for your time and attention today.